It is my pleasure to now introduce Hiranya Sudasinghe. Hiranya received his bachelor's degree in zoology from the University of Peradeniya, graduating with a first-class honors. He then followed a master's and is now presently reading for his PhD at the Institute of Ecology and Evolution at the University of Bern in Switzerland. His doctoral project is on understanding the genomic consequences of miniaturization in zebrafish relatives. He has lead authored some 24 scientific papers, principally on the phylogenetics and taxonomy of Sri Lankan freshwater fishes. He's a co-author with Rohan Pettiagoda of the upcoming book, The Ecology and Biogeography of Sri Lanka, a context for freshwater fishes. And for the attendees of the event today, there's a special pre-publication offer for this book that has been extended till the 21st of November. I will put the flyer and the details for this shortly in the chat box. So over to you, Hiranya.
Thank you very much, Zainab, for that introduction. Let me share my screen. Can you see the screen?
Yes, I can.
Okay. Okay, so hello everyone. Let's dive into the world of freshwater fishes in Sri Lanka. Let me just give a brief intro about me. I've always been fascinated by nature since I was a kid. And then I have been a member of YZD, the Young Zoologists Association. And then I decided to follow my passion and do a degree in biological sciences. But I've been fascinated by nature since I was a kid, and that's how I ended up doing biology and doing what I like to do. So first, to give you a general introduction on Sri Lanka, I mean, this is like most of you probably already know, but this may be relevant for our discussion on the freshwater fishes. So, you know, in Sri Lanka, the rainfall is governed by the monsoonal system, and we have the southwest monsoon and the northeast monsoon. So the southwest monsoon, which brings rain to the western quadrant of the island, receives rainfall of more than 2,500 millimeters a year. And this is the region that we identified as the per humid region, which means a region in which the average monthly rainfall remains above 100 millimeters, even during the dry season, which could last about 2 to 3 months. So this is the same region in which you find the rainforest habitats, in which you see the highest, the rich endemism of both flora and fauna. This is also, unfortunately, the region in which there's highest population density, and therefore the most threatened part of the island in terms of biodiversity loss. Then when we discuss about the river basins, we have 103 river basins in Sri Lanka, originating from the Central Hills and the Knuckles Hills, and they radiate in a circular manner throughout the island. So with this diversity of climate, habitat, heterogeneity, there are various different types of freshwater aquatic habitats. For example, you find these streams and rivers in the rainforest, which harbors a very rich endemic number of freshwater fishes. Then you also find dry zone rivers such as the Mahaveli River, which is the largest river in Sri Lanka, and also riverine dry zone habitats. In addition to these streams and rivers, you also find man-made reservoirs, mainly in the dry zone, and also lowland floodplains, both in the dry zone and wet zone. So all these different types of habitats, they harbor a variety or a diversity of different species of freshwater fishes. In addition to these, of course, you also find the man-made canals and streams, which in addition to streams traversing plantations, which also in fact harbor some freshwater fishes. So there are about 100 species of freshwater fishes in Sri Lanka that obligatory depend on freshwater at least for part of their life cycle. And among these about 100 species, more than 50% are endemic to the island. And these freshwater fishes are represented by around 22 families. And in addition to these native species, we also have around 25 to 30 alien species, which are now naturalized in the freshwater system in Sri Lanka. There are two endemic genera of freshwater fishes. One is the genus Malpuluta, which is a monotypic genus, which means there's only one member of this genus, and it is only found in Sri Lanka. And this is Malpuluta cretseri. It's now a rare species confined to a few locations in the wet zone. And there's also another endemic genus of freshwater fishes in Sri Lanka. This is the genus Rasboroides. This is represented by two species. One is Rasboroides pallidus, and the other is Rasboroides vetrifloris. Both these species are confined to the rainforest habitats in Sri Lanka. And if you just go through some of the freshwater groups in Sri Lanka, we have the family Angulidae, which is represented by two species of eels belonging to the genus Angula. And in Sinhala, we call these eels as Ando. And we have two species, and the special feature about these eels is that they are catadromous. So catadromous means the adults migrate to the oceans to breed, and the larvae migrate back to the freshwater, and they grow in, they have most of part of their life cycle in the freshwaters. Then we have the family Chanidae, which is represented by the snakeheads of Sri Lanka. We have about 5 species in the island, and the largest endemic freshwater fish in Sri Lanka is a member of Chanidae. It is known as the giant snakehead. Now we recognize 2 species of giant snakehead in Sri Lanka. One is Channa marulius in the northern dry zone, and then we have Channa ara in the Mahaveli Basin, and another population in the southwestern wet zone. Then we have catfishers of belonging to 4 families. We have the catfishers of the family Bagridae, which we usually call as Ankutto, and we have 4 species of them in Sri Lanka. Then we have family Claridae, represented by a single species, Clarius brachisoma, which we usually call as Magura. We also have another catfish belonging to the family Heteroneustidae, species known as Heteroneustus fossilis. And this is what we normally call as Hunga. And the special feature of this catfish is that in the pectoral fin of this species, there's a venom gland attached to it. So if you get stung by stinging catfish, it won't be a pleasant experience. Then we also have the family Siluridae, which is represented by 2 genera in Sri Lanka, the genus Ompok and the genus Wallago. And the largest freshwater fish in Sri Lanka belong to the genus Wallago. The species is Wallago actu. We call it as Valaya in Sinhala, and it can grow to a size of about 1 to 1.5 meters in length. Then there's another interesting group of fishes which are known as gobies or valigobo in Sinhala. So we have around 7 species, and some of these gobies are known as Cicidine gobies. So in this case, just like in the eels, but in these gobies, the adults live in freshwater and they spawn in freshwater, and their eggs might get washed away back into the sea, and the larvae migrates back into the same habitats as the adult gobies, which are found in the freshwaters. We also have another interesting group of eels in Sri Lanka. These are known as blind eels because they have a very minute eye, and they are usually found in swamps and habitats which include a lot of mud, and they are very difficult to discover in the field. And we have two species. One is Ophitis disilvi, and the other is Ophisteron bengalens. We have another group of fishes belonging to the family Mastacembelidae, which we usually call as Telio. There are two species of this. One is Mastacembelus armatus, which is quite a common species, but we have another species which is actually very rare, which is known as Macrognathus penthophthalmus, and it is known as Buttercollothelia, and at present we are not exactly sure if there's an extant population of this species, or we do not, because it had been at once a very common species, but now it has become very rare for reasons that we do not understand why exactly. However, if you look at the freshwater fishes in Sri Lanka, the diversity, the majority of the freshwater fishes are represented by the order Cypriniformes. So the order Cypriniformes is one of the most species groups of fishes in the world, and the highest diversity is observed in Asia. In Sri Lanka, this order is represented by 18 genera belonging to 4 families. These 4 families are family Corbitidae, which is represented by a single genus of Lepidocephalichthys. Then the family Nemacalidae, represented by 2 genera, Cistura and Paracanthocorbites. And both these families, the members of these families are generally known as lochos. They are benthic species found in the bottom of an aquatic habitat. Then we also have the family Cyprinidae, which is represented by barbs and carbs, as we generally call them. And then we have the family Darnionidae, which is represented by Darnios, Rasboras, and small species of cyprinids. So some of these freshwater fishes show an insular diversification in the island. So what is an insular diversification? These where a common ancestor enters Sri Lanka during the past, and then it diversifies into different species within the island. For example, we see such diversification in groups like Devario, which is represented by 5 species in Sri Lanka. 4 of them are endemic. We have a genus Labuca, which is represented by 3 species. We have genus Rasbora, which is represented by 6 species. And these cypriniforms they highly differ in the body forms, their sizes, habitats, and life history traits. For example, the smallest freshwater fish in Sri Lanka is a member of the genus Horodandia, which is the species Horodandia tucorali, and it is only it can reach only to a size of about 20 to 30 millimeters in standard length. Whereas the largest cyprinid in Sri Lanka is Tor kudri, which we call as Lehella or Horopolea, and then this could actually reach a size of more than 60 centimeters in standard length. There are also some species which are narrow range endemics. So a narrow range endemic is a species which is confined to a single locality, or maybe confined to a single river basin. For example, we have a species called as Cystomus asoka, or perhaps you may have heard of the species as Asocopethia, which is found only in few locations in the Kalani basins in the Dharaniagala and the Kitulgala area. We also have another species called as Pethia bandula, which is confined to a single location in the headwaters of Kalani Basin in the region of Galapitamada. Another example is Devario patirana, or we call it as Patirana salya, which is only confined to a single river basin in the southwestern wet zone. It is only found in the rainforest habitats in the Nilwala Basin. So if you look at the species accretion, how the number of species have changed over the past few decades or during the past few centuries, the number of species had been continuously increasing. And if you compare it also with reptiles and amphibians, since the early 1990s, there was a lot of work on exploration of these groups. And as a result, there was a steep increase in the number of species in the island. And if you want to read about freshwater fishes in Sri Lanka, I would suggest this book by Mr. Rohan Petiagoda in 1991, which is even today, though some of the facts are outdated, if you want to start reading on the freshwater fishes in Sri Lanka, this would be the best starting point for you. So if you look at the research on freshwater fishes in Sri Lanka, the research in the past focused mainly on taxonomy, ecology, and fisheries of the different groups of fishes in Sri Lanka. And when you mean traditional taxonomy is mainly using morphology as a criteria alone. However, now there's a shift from traditional taxonomy, and now we also try to understand what are like the evolutionary relationships and the phylogenetic affinities of the Sri Lankan freshwater fishes. So what do I mean by evolutionary relationship and phylogenetic affinities is trying to understand how closely related different organisms in their evolutionary history. We also try to look at the phylogeographic structure and patterns of diversification in the Sri Lankan freshwater fishes. So the idea of phylogeography is to understand the patterns and processes that may be responsible for the geographic distribution of a species by using genetic data. So we've been using a combination of different criteria. We've been using morphology, and we have also been using molecular data, both mitochondrial DNA and nuclear DNA, and also comprehensive geographic distribution to delineate species boundaries in a general lineage concept of a species. Now you may wonder what a general lineage concept of a species is. Well, if you define a species, it is quite a debatable topic. We all know the biological species concept, but in the world of speciation, there are a lot of definitions of what a species is. And when you try to read more about species and when you try to understand more and more, it becomes complicated. So the idea of general lineage concept is that you use different criteria to define, or based on different criteria, you come into a conclusion of what we define as a species in this context. This is important because a coherent understanding of the identity and the distribution of a species is the starting point for the conservation management. So I've been doing field work with my colleagues for some time now, and the points in blue are the locations in which I have been where I've done field work, and also we have a very good reference collection in the museum of the WHT collection, and as a result of all this data compiled together, we now have a fairly good understanding of the distribution patterns of most of the widespread species of freshwater fishes in Sri Lanka. So the fieldwork usually include recording ecological data, recording habitat data, taking field notes, making natural history observations, and also, if possible, making underwater photography. And based on this fieldwork, the data collected during fieldwork are also important when we go back to analyzing our data, which we collect in the lab, which include examining specimens in the museums and also generating genetic data using several molecular markers. So as a result of this work, several publications have come out. Some of these are based on primary taxonomy, which means either clarifying the taxonomic identity of a species or either describing a new species of freshwater fish in Sri Lanka. However, some of these studies have also resulted in a much more broader subjects such as systematics, phylogeny, and phylogeography of some Sri Lankan freshwater fishes. And these are perhaps the first ever approach to explore this kind of relationship using molecular data in the freshwater fishes in Sri Lanka. So as a result, we have now some understanding of different patterns and processes of freshwater fishes in Sri Lanka. So I thought I would take 3 examples in this talk and then show what kind of work I've been doing in the Sri Lankan freshwater fishes. The first theme would be a taxonomic revision. And for this, I would take the example of the genus Labuka. So there was a taxonomic revision of this group, and as a result of that, there were 4 species identified. They were Labuka varuna, Labuka ruhuna, Labuka lankensis, and Labuka insularis. All these 4 species were considered as endemic, and this study was based on morphology alone. And among these 2 species, Labuka lankensis showed a distribution in the dry zone, and Labuka insularis was defined as a species confined to the Knuckles Hills of Sri Lanka. And these 2 species were differentiated by Labuka insularis possessing a longer pelvic fin compared to Labuka lankensis, and also by having a short snout compared to Labuka lankensis. So in my work, I sampled around 56 locations representing 14 of the island's principal river basins. And I also examined morphology of the specimens, and I also analyzed both mitochondrial DNA and nuclear DNA of this group in Sri Lanka. So this is Labuka insularis. It is usually found in these kind of fast-flowing habitats in the Knuckles range. And you can see compared to Labuka lankensis, which is found in more riverine dry zone habitats and also sometimes in reservoirs in the dry zone. In Labuka insularis, it has a shorter body depth compared to Labuka lankensis and also a much more longer pelvic fin compared to Labuka lankensis. So this was how these 2 species were diagnosed based on morphological data. So this was the original based on the holotype, which is the original place from where the species was discovered. Labuka insularis from the Knuckles Hills and Labuka lankensis from lower reaches of the Maha Valley. However, both these belong to the same river basin. So in my study, I sampled, in addition to the Maha Valley basin, I also sampled some other dry zone river basins, such as the northern dry zone basins and also eastern basins. And the outcome of our study was quite interesting. So this is a phylogeny or a reconstruction of the evolutionary relationship of Labuka in Sri Lanka based on the genetic data. So actually, I also showed that Labuca varuna and Labuca rufna, which was only found in the wet zone, in fact represent a single species. We also found a new species of Labuca from the eastern basins, like in the headwaters of eastern basins, such as in the headwaters of Galway Basin, which we named as Labuca hema. However, in the case of Labuca lankensis, the pattern we saw was not that what we expected. So what we found was that in Labuca lankensis group, that what was identified as Labuca insularis here in red and Labuca lankensis from the dry zone, they all form a single clade, which actually represent all individual samples from the Mahabali Basin. In addition to that, it also formed two clades, one representing an eastern clade which represents samples coming from the eastern basins such as Walave, Kumbukwan, Manik basins, and also another clade representing samples that come from the northern basins such as the Daduroya, Malwatoya, Kalaoya. And when we also analyzed the morphology of specimens using a much more larger sample size, we also found that in the case of Labuca insularis and Labuca lankensis, there was a clear overlap of the characters which was earlier used to differentiate these two species. So it looked like the morphological differences in this case, which was earlier observed based on a limited sampling size, was a result of plasticity of this group based on the different ecological or environmental habitats in which these populations are found. I'll take the next theme of my talk, which is the discovering of a new species. I would take the case of Devario. So Devario is quite a ubiquitous group in Sri Lanka, especially Devario malabaricus, which is found in a wide range of habitats. And it is among the 4 species, Devario malabaricus is also found in India, whereas the rest of the species are endemic to the island. So we have Devario malabaricus, a widespread species found in addition to Sri Lanka, also in Peninsula India. We have Devario monticola, a species confined to the highlands around Agrapatana area, in which the elevation is about 1,500 meters above sea level. We have Devario patirana, which is confined to the Nilvala Basin, to the rainforest habitats of the Nilvala Basin. And then we have Devario micronema, which is confined to the rainforest habitats in the southwestern wet zone from Kalani to Dim basins. So I examined the morphology of specimens, and also I examined the genetic data, both mitochondrial and nuclear DNA. And as a result of that, I found a new species of Devario, and this was discovered from the headwaters of Maoya Basin in the Aranaka area. The discovery of this one was quite interesting. So I found this species first in 2015 while I was an undergraduate in university. So I had a habit while I was in university, I used to travel a lot with my friends in universities to their homes and just explore every stream and river found in their area. So this was actually in Aranaka, where my roommate lived at that time. So I went to his place, and then I found this fish actually just next to his house in the headwaters of the Maoya Basin. So I named this species as Devario memorialis. It took some time from 2015, finally managed to publish this in 2020. And the reason why I named this species was because during the same time, while I was doing fieldwork during this area, there was a major landslide, maybe you may remember, in which there was a lot of people unfortunately died because of this landslide. As a result, I named this species in memory of those people who died during this landslide in Aranaka. If you look at the phylogenetic position and also the morphology of this group, it is quite interesting. It is the sister group. The rest of the wet zone species of Devario in Sri Lanka to Devario pathirana and Devario micronema. And also, if you look at morphology, it is very clearly distinct from rest of the species of Devario in the island. It is also interesting to note that while I was doing underwater observations, I also noticed that both the new species, Devario memorialis, which are here, and also, in fact, Devario malabaricus, they actually occur in the same habitat. So it is also possible that there's hybridization ongoing between these two groups, but we do not have any evidence to support whether there's hybridization or not in these groups of fishes in the type locality of this group. Now I will take my third theme of my talk on the phylogeography of freshwater fishes in Sri Lanka. That is, to understand the patterns and processes of the distribution of species by using genetic data. We are coming to an interesting pattern that we observe in the freshwater fishes in Sri Lanka. For example, if you take a species like Devario micronema, which is a kind of a habitat specialist, it's found in these kind of rainforest habitats in the southwestern wet zone. So among the species of Devario, among the endemic species, Devario micronema is the species which show most widespread compared to other endemic species of Devario. It is found in the rainforest habitats in the southwestern wet zone. So when we analyze the genetic data, what we see is that there is strong phylogeographic structure, even between the adjacent basins, which means the haplotypes. So in this case, a haplotype is a representation of unique set of sequences which are analyzed. So the haplotypes between river basins are not even shared, even when they are adjacent to each other. And this may suggest that there is restricted gene flow among these habitat specialists which are found in the rainforest habitats in Sri Lanka. However, compared to this, if you look at a widespread, a generalist species like Devario malabaricus, which is found in all sorts of habitats from lowland floodplains to the central hills, it doesn't show any clear phylogeographic structure. The haplotypes from the dry zone and the wet zone, and haplotypes from the lowland floodplain and the central hills, they are all shared between each other. So this may suggest there is unrestricted gene flow in the habitat generalis like Devario malabaricus, whereas there is clear phylogeographic structure in species which are confined to rainforest habitats like Devario micronema in Sri Lanka. A similar case we also see in Labuka varuna. Again, Labuka varuna is confined to the southwest wet zone. It is found in basins from Attanai Galu to Nilwala basins. And again, based on the sample, based on the data analyzed so far, we do not see any haplotypes shared between even the adjacent basins, which may suggest there's a restricted gene flow and strong phylogeographic structure in Labuka varuna. However, compared to Labuka varuna, if you look at Labuka lankensis, what we see is a different picture. In the case of Labuka lankensis, we find three different haplogroups in Sri Lanka. One haplogroup represents the samples that derive from the Mahaveli Basin, from this region, and another haplogroup which represents samples from the northern dry zone, from basins such as Daduru, Kala, and Malwatu, and another haplogroup which represents samples from the eastern basins such as Galloya, Kumbukan, Mahaveli, Manik, and Mangalavatan, Aru basins. These are the basins analyzed so far, but this may be true even when we analyze even further more data. So the reason why we do not see the haplotypes between the lowland floodplains of Mahaveli and northern basins such as the northwest subclade, which derive samples from Malwatu and Kala. Why haplotypes are not shared between these groups is not yet clear. But in the case of Labuca lankensis, and also in another species like Petia melanomaculata, we see this pattern of 3 distinct haplogroups representing their geographic distribution in the dry zone of the island. Let's also look at another interesting group of fishes, which is Gara silonensis. So rheophilic fishes are species which are adapted to fast-flowing habitats in the island, in the aquatic habitats. For example, such rheophilic fishes commonly occur in the rivers of southern India. For example, there are many species of lochers and even catfishes which are adapted to these fast-flowing habitats. Unfortunately, we do not have any such rheophilic fishes in Sri Lanka, except for only one group, which is Gara silonensis. It is a species found in both the wet and dry zones of Sri Lanka, wherever there's a habitat of fast flow and rocky bottom. So I sampled around 38 locations representing 16 of the principal river basin. And this was also a collaborative project with our colleagues from India. And as a result, we also had data from 36 locations in Peninsular India. We analyzed the mitochondrial DNA, and we also analyzed the nuclear DNA of this group in Sri Lanka and in Peninsular India. Now, the interesting thing on Gara is that the lower lip of Gara silonensis is modified as a sucker so that it can hold on to the rocky habitats in the fast-flowing habitats. So when we analyzed the data set, we found some interesting patterns. We found strong phylogeographic structure in Gara silonensis, which represent different groups which are close to adjacent basins. And an interesting thing that we found here was that one would expect that the ancestral species that enter Sri Lanka from Peninsular India, from Malwatu and Kala basins, should be the basal group within Sri Lanka, which means it should be at the basal position of the phylogeny compared to the rest of the species in Sri Lanka of the different other populations. But what we found was, in fact, the population of Malwatu and Kala of Gara that we find today are in fact derived from, they also belong to the same haplogroup as that from the Mahaveli Basin. This may perhaps suggest that there were several extinctions in the past that extirpated the dry zone populations, such as the ancestral Gara in the Malwatu and Kala, and later a population of Gara from the Mahaveli Basin dispersed back to the dry zone basins such as Malwatu and Kala. This also suggests there had been some historical connectivity between Mahaveli, Malwatu, and Kala basins. And we have now also found evidence for this and by using Dawkinsia as an example, where the example of Dawkinsia srilankensis, which is found only in the Mahaveli basins in the Natas region, and its sister species, Dawkinsia tambraparani, is found only in the Tambraparani River in the Peninsula India. And then we also think there were several extinctions, one or actually several extinctions even in the wet zone, and in which the populations of the rainforest habitats acted as refugia, in which the species again dispersed back and diversified in the island. However, there are some limitations of these studies as well. One major limitation is the lack of comparative data, both in terms of genetic data and in terms of specimens from southern India. And we really need these data to understand a clear picture on the species in Sri Lanka. And these studies are actually only also based on fewer molecular markers, whereas like few mitochondrial DNA markers and a single nuclear marker, whereas nowadays we use a lot of several molecular markers, and that may actually give much more clearer picture on the evolutionary relationships of these groups. So these studies actually only provide first insights into the patterns and processes, but it's really difficult to make any strong conclusions out of these. However, there is also some significance of these studies. For example, many new species of freshwater fishes are described from South Asia each year. Or this is also really based on limited sampling, either based on a single location and also based on just using a single molecular marker. The understanding the evolutionary relationships within these species are, however, still at their infancy. So in this context, the studies I've been doing in the freshwater fishes in Sri Lanka are perhaps the first ever approach to explore such evolutionary relationship using molecular data in the Sri Lankan freshwater fishes. Right. And one important thing was using phylogeographic sampling, so that we consider both intra-specific variation and as well as inter-specific variation, which is really useful when you try to understand the diversity of a group for any group actually. And we analyze the species boundaries using a combination of criteria. We use molecular data, we use morphology, and we also consider the geographic distribution before coming into conclusions of defining what our species are in our studies. And as a result, we clarified the taxonomy, which I would think is the starting point for conservation planning and management for any group of species. As Zainab mentioned before, so some of this work, especially the work we did on the phylogeography of some of the groups, we actually synthesized this into a book with Mr. Rohan Petyagoda and myself. And the book is actually on the ecology and biogeography of Sri Lanka by taking various threads of geology, ecology, biogeography of different groups of both flora and fauna. And then we put a special focus on the freshwater fishes, which is what we have been studying mainly, and for which we think we have quite a good data set that we can, based on which we can make some interesting patterns on the freshwater fishes distribution in the island. So if any of you are interested to understand or to read on the ecology, biogeography, phylogeography of Sri Lanka, I think this would be, you would find this quite interesting read. I would like to thank my two mentors, Mr. Rohan Petyagoda and Professor Madhav Kumbura, for mentoring me throughout my career, and I was quite lucky to meet them when I was really starting with my work. I would also like to thank the Department of Wildlife Conservation and the Forest Department for providing all the necessary permits and allowing for our work. And for my university, where I did my bachelor's and my master's, University of Peradeniya, for providing all the facilities and all the great lecturers in which I learned a lot. And for all my different groups that provided me funding to carry out my work in Sri Lanka. And finally, to all my lovely friends, with whom I enjoy a lot of fun field explorations and traveling throughout Sri Lanka looking at fish. Thank you very much.
Thank you, Hiranya, for that fantastic talk. It's amazing to see the depth of work you have done in regards to fish in Sri Lanka. To get started with the Q&A, I was just going to start by asking about, you mentioned at the start a little bit about invasive species and alien species in the freshwater ecosystems. What is the impact of these invasive species on native species, on the indigenous species in the system? And also, would you be able to elaborate a little bit in terms of how they enter the freshwater streams and rivers of our country?
Yeah, so I think when we talk about the alien species, if you look at the origin, there are several modes of origin. One major origin is through aquarium trade. There are some species they're available in the aquarium trade, and then for some reasons they get naturalized in the freshwater system. And there's also species which have been introduced as a result of fisheries and aquaculture, and they are like now one of the most commonly found species in the dry zone reservoirs. For example, if you take tilapia, it is one of the commonly encountered fish in the dry zone rivers reservoirs. But when you need to evaluate when a species is really invasive or what kind of impact it has, then you really need to look at the biology, ecology of this group, and then also to compare if there are competing groups of native species in Sri Lanka as well. For example, there are some species which have been introduced through the pet trade, such as the knife fish, which is found in the dead zone lowlands, which is a carnivorous species, and that would be a major impact to the small native species in Sri Lanka. Now, we also see a pattern of snakeheads, which is very popular in the world and also in Sri Lanka. There are many different types of snakeheads, and there are some really very predatory big snakeheads also really easily available in the pet trade in Sri Lanka. There had been some records of these also found from the native system, but we are not exactly sure whether there's a reproducing or extant population of this group now established in Sri Lanka. But if those predatory species now established in Sri Lanka, that would be a major impact to the native species. Another example is the tank cleaner. Most of us know we keep this fish in the tanks, and then it becomes big and not so attractive as it used to be. And then you put them into the native, the rivers and river reservoirs. And this is actually a major pain to the fishermen as well, because when these tank cleaners get entangled in the nets, and it is very difficult to take them out from the nets as well. So I think in terms of impact for every different species, the impact need to be evaluated separately. But I think most of the impact is from the big carnivorous species of exotics, which are now established in Sri Lanka.
Okay, there's a question that's coming. Can you elaborate how these new species impact conservation measures, especially if they are range restricted? I assume they're referring to newly discovered or recorded species, and how do y'all come about with the conservation measures to protect them, and especially when they're range restricted?
Yes, so I mean, the interesting thing in Sri Lanka is there are a lot of narrow range endemics. It's not just the new species described, but I mean, if you look at it, even the species that had been described like decades ago, like Asoka Ba or Devario Patinana, it's still a narrow range endemic. So it's more like what kind of, it doesn't matter if it's a new species or if it's a species that had been described before, but what kind of measures we need to take. And I would think one important thing is really to understand the biology, the ecology of a species. For example, we know Asoka Ba is found only in 2 locations in the Karenni Basin. It's restricted to Kitulgala and Dharanigala area. That's it. But I don't think anybody knows how this species reproduce. We do not know of the breeding biology of this species. So definitely, in terms of conservation, you really need to understand the breeding biology, and you really need to do in situ and both in situ as well as ex situ conservation. And also, if you take the example of Petia bandula, the bandula bar, we all know in our textbooks, in our A-level syllabus or the O-level syllabus, we learn about this species. It's found only in a single location. And IUCN did a great job in really making the community aware of this group in the region, in the places where they are found, and so there was a lot of community awareness, and was really a job well done in terms of bandula barb conservation. But you really need to understand that if this species get extinct in this region, which could be a result of a stochastic event. The thing on fishes is that, especially in freshwater fishes, any stochastic event can cause an extinction or like major disruption of the population. And also, we do not know whether these narrow range endemics are really going through a bottleneck in terms of their genetics. So we do not have any population level data on this narrow range endemics. So we really need to study them in much more finer detail and really look into the population level data. For example, we recently worked on Petia in Sri Lanka, and what we found out was that Petia bandula, based on the genetic data which is already available, it's found within Petia nigrofasciata. So does that mean Petia bandula and Petia nigrofasciata is same? It is difficult to make a strong conclusion, because you have to understand also that now Petia bandula had been undergone through a strong conservation action. So there has been a lot of conservation plan on this. So we, as a scientist or as a taxonomist, when you need to make a taxonomic implication, you also need to understand much more on the conservation scenario of this group. And also you have to realize that the limitations of the data in which you analyze the data set. So you should always need to be aware of the limitations and then make some conclusions on these groups.
Thank you. There are quite a few questions that have come in regards to aquarium trade and freshwater fish in captivity. So there are people asking about how do they get the fish in aquariums? Is it legal to capture invasive species? So would you like to share your thoughts on the aquarium trade, and maybe if you can give us some insight in terms of what are the legal protections that freshwater fish have in the country?
Legal protection of the aquarium species that are now naturalized in Sri Lanka?
Or just in general, especially in regards to invasive species, are you allowed to capture them for aquariums? Is one of the questions.
I'm not exactly sure whether, like, once it is naturalized in Sri Lanka, whether you can catch them or not. For example, I mean, you have let's take the example of trout, which had been introduced in during the colonial period in the Hidden Plains, and because now Hidden Plains is a national park, we are not allowed to catch any fish. But if there's a aquatic, like an aquarium species now naturalized in Sri Lanka, whether you are allowed to catch them or not, again, for the aquarium trade, I'm not exactly sure actually is that allowed or not.
Yeah, that's fine. I suppose there might be some protections in regards to the Fauna and Flora Protection Ordinance, but I'm not sure in particular to the species. There's a question. We have many trans-basin water diversion projects taking place over the recent past, such as from the Ambanganga to Mahavali and from the Umaoya to Kirindoya. What effects have these diversions had on freshwater fish distributions?
That's a cool question. So, so when you do a trans-basin, there's definitely mixing up of population. For example, let's say a common species like Devarium malabaricus, which is found throughout the island. And then we found that in that case, there's no phylogeographic structure in this group, but that is only based on mitochondrial DNA markers. So, but when you do a transbasin activity, then there's definitely mixing up of the rivers, and then species, the different population would disperse throughout. And then of course there could be hybridization between even different species. For an example, some species of Rasbora, like Rasbora naxi and Rasbora microcephalus, closely related, but they could hybridize. Whether we can detect that kind of hybridization it depends really on what kind of data you analyze. For example, the different genetic data you analyze, they have the rate of evolution of the genetic data is really different. So for example, if you analyze mitochondrial DNA, for mutations to occur in mitochondrial DNA would take a longer time. So if this transbasin interaction took place, let's say 5 years ago, you may not really see a pattern of hybridization in this group just by analyzing mitochondrial DNA. However, if you analyze some other data like SNPs, like nuclear, like markers such as fast evolving single nucleotide polymorphisms in these groups, actually you might actually see there's hybridization going through these groups. Yeah, so it really depends on the type of data you analyze and whether you would see it, whether you would detect such hybridization. But definitely when you do transbasin activity, there would be really mixing up of different populations.
Interesting. Are there any good pocket guides for freshwater fish available? I know you mentioned about the book that Mr. Rohan Petiagoda had done, but are there any pocket guides out there?
I think there's a book by Mr. Sampath Kunatilaka that was in 2007. I think that's in terms of pocket guides. I think that's the only pocket guide we have on freshwater fishes. But then we have the book by the Madhura De Silva and Nadika Hapwarachi and the group, that they also have a freshwater fish book, but it's not a pocket guide. Actually, it's also like the book by Mr. Petiagoda. Yeah.
There are some, I think, young people on the talk, and they're asking about how they're keen to study their masters and study freshwater fish, and they want to know how they can get involved.
Have curiosity and the drive to do work demanding. Sometimes the work could be demanding, but I think if you have the curiosity and the drive, you can do it. But in general, I would advise, if you like to do, let's say, if you're interested in ecology, if you want to do ecology, nowadays just having an interest itself won't be enough. You really need to learn a lot in terms of, for example, if you really are into ecology, it may be really worthwhile giving some effort to start learning a really good statistics package, like you really need to work on those. And if you are really interested in evolutionary biology, and if you really like to work with molecular data, I would suggest you start programming, learn a little bit of programming from the very early age. Even if you're undergrad, really start concentrating on those, because the data becomes bigger and bigger, but the computation powers become more and more difficult. So really you need to put some effort on that. Yeah, but I think you can also join the societies like YZA, which does a really great job. And you can really get an overall idea of the system on Sri Lanka. And then I guess, yeah, you can also join the monthly talks like organized like here. And yeah, I mean, it depends really on what kind of background you are in, but really what really you need is the interest and the curiosity.
Yeah. Climate change has affected the distribution of fish in Sri Lanka over the years, over the course of history as well. But how is the current changes in climate, and how do you foresee that impacting upon freshwater fishes on the island?
Yeah. So as I said before, like in terms of freshwater fishes, the the even a stochastic event can cause a major impact if you look at a narrow range endemic. So in terms of climate change, the impact on that, we really need long-term monitoring, both at the population level of a species as well as long-term monitoring of climate within different regions in the island. For example, as far as I know, the population level studies in Sri Lanka, not just for fish, but I don't, maybe perhaps most of the groups is really like we are still in a more like a macro evolutionary level, but we really do not look at the population level data, for example. But that is, I think, really important when you really want to make strong inferences or strong conclusions, especially when it comes to conservation planning, as well as when it comes to climate change. We really need to do long-term monitoring, and for those narrow-range endemics, we really need to build up a strong conservation plan based on, for example, understanding their breeding and ecology, biology of those species.
I would just want to encourage people, if you have questions, please do send that into the chat box. And if you can send that directly to me, that would be great. There's a question about that you study many different species of fish you have studied. What is your favorite you have studied so far?
Favorite, I would say I like the barbs, the cyprinids a lot. But I mean, I don't think I really have like a favorite in a way, but yeah, it's all fish I like, I would say.
Okay, there's another question. What are the species of freshwater fish, if any, that have gone extinct in Sri Lanka?
Extinct. Okay, so there's one species, or rather two. It's a genus of Parelius. And this group is found in India. And it's very similar to a species of Devario, but it's a different group of fish. And Frederick Kellard in the 19th century, he mentions of two species from like Nuara Elia or the Central Hills, and that he says that, and one of the scientists from India who was one of the major ichthyologists in South Asia, known as Francis Day, he mentioned in his description that he also recorded this species from Ceylon, and Frederick Kellard mentioned this in his book from Nuara Elia area. But that's the only record. We do not have any specimens, like historical specimens of this species, and we really do not know whether this species really occurred and that it got extinct, maybe as a result of the rainbow trout, or maybe as a result of clearing of the Central Hills for tea plantations. We really do not know, but that is one record of a species that had been mentioned in the 19th century, but now we have no trace at all. But there are some other species, for example, now Labio lanke, which was known as Tabelea, and it was a common species like a few decades ago, but starting from 1980s, it was found to be very rare in the wild. And we also thought it may be perhaps extinct or potentially extinct in the island. But then we actually found this species in the northern dry zone, in the Malwatu Basin and in the Kala Basin. This species is actually there, and it is found in several locations in this region as well. There's another species of a spiny eel I mentioned before. It's called Macrognathus penthophthalmus, which is called Batacolatelia. Again, a common species a few decades ago, but since 1980s, it has become really rare. We do not really know why. My suspect is there had been scant records of this one from few locations even now. So it is, I think it's not extinct. It is there, but I think maybe we are sampling or we are looking at it in the wrong place. The habitat could be different, so we are missing the habitat of this species, so we are missing any records of this species. Another record of a species is a goby called Stiffodon martinsteini. So this species is based on a single specimen which had been collected in the 1980s from the ichthyology area. And this was described in late 1990s. And this is another Cicidine goby. So it has a life cycle in which the adults live in freshwater, and the larvae, the eggs goes to the ocean, and then the larvae come back to the freshwater. But we have never recorded, at least as far as I know, nobody has recorded this like an extant population of this species in Sri Lanka. And every time we go snorkeling in ichthyology or any area in this area, we all tell to our friends and all, now we are going to see a Stiffodon martinsteini in this habitat, but so far we have never found that. But maybe it's real because if you look at the distribution of Stiffodon genus, it is found in these oceanic islands and most of the oceanic islands and in Southeast Asia, but Sri Lanka is like towards the edge of its distribution. So it could be actually maybe the extant population of Stiffodon martinsteini is not found in Sri Lanka, but somewhere else, in Andaman Islands, so I don't know, maybe somewhere else. But it was just one record of a specimen from Sri Lanka, but we have not recorded it so far. Yeah.
Fascinating. Are there any good citizen science programs for freshwater fish that you know of? It's ongoing in citizen science work.
I think if you are in iNaturalist, I think that would be a best option. I think if you are interested in citizen science, to get involved, because it helps you with identifying a species and also being involved in this kind of activities. But as far as I know, I'm not exactly sure whether in Sri Lanka there are a lot of citizen science programs. But I mean, if you join a society like WNPS, YZD, I guess there are a lot of citizen science programs in which you can get involved. There's also a question asking whether Gymnothorax polyrundum is found in Manikganga. I hope so, but I do not know. But so it's another species which is like it's another eel, which, if you look at the natural distribution of this group, this genus or the species, again Sri Lanka is towards the edge of the distribution of Gymnothorax polyrundum, and there had been several records in the past. Dr. P.P. Dharanegala recorded some of those from Kalu Basin, but we really do not know whether there's an extant population of this world. These are all like rare records. So we really do not know much about this group, and I think these are really difficult to find as well, because these are found in really deep waters, and you really come across them if you through a fisherman or if you really focus on catching one of those. It's not a species that you would just snorkel and come across. Yeah.
There is a question asking how many endemic fishes are in the knuckles. I don't know if you'll be able to answer that. Maybe you can also elaborate about river basins that have a very unique composition of species and that stand out in Sri Lanka.
Yeah. So if you look at knuckles itself, there are a few groups, a few species which are kind of confined to the region, but not exactly like limited in terms of distribution. For example, if you take the species Dawkinsia srilankensis, which is known as Malpathia, and we thought it is found only in few habitats in knuckles range, but what we now see is it is found in a region of the whole knuckles range, and gets as far as the Angamadilla area close to Polonnaruwa, but which is still part of Ambanganga. And there's also species like Cystomus martinsteini, which we thought earlier has really confined to the Knuckles region, but it is much more distributed within these tributaries within the Mahaveli Basin, such as Ambanganga, Kaluganga of Mahaveli, etc. There's also a labial fishery, but it is not really confined to Knuckles, but it is more like confined to the central hills and Knuckles of the Knuckles of Mahaveli. Yeah.
All right. I've seen major changes in nomenclature in species names. I think they're saying about the Petia. What are the factors that determine these changes, and is it subject to further revision, hence left fluid depending on further scientific discovery? Do these inconsistencies have an effect on the community who depend on nomenclature for identification purposes?
It depends, I think. I mean, science progress every day, right? So when facts changes, our idea and our science also changes. So there's no hard and fast rule of species and say this is it, and then we don't want more changes. Then it doesn't become science; it becomes something else. So when science changes, when more data, if we have more data, more understanding, different techniques, we see different patterns, and that's how science progresses. If we stick to what was like in the 19th century, we would all still like sometimes call all these species in one name. I mean, nomenclature is really a facility to make understanding of biodiversity, right? It's just provide you a platform to understand what a species is, but I don't think it would necessarily cause any really major impact on conservation, because what you really conserve is it's not a name of a species. You conserve like that population, and so it really doesn't matter if a name changes. I mean, what I would think is that I feel sometimes we in Sri Lanka, we are too much attached to the names of a species, but it really doesn't matter. You really conserve a population, or you really conserve a biological entity. And taxonomy and nomenclature is more like a platform to make it easy for us to understand the biological system. Yeah.
Absolutely. Understanding more about the systems and the threats affecting these species are what's, I suppose, key for conservation purposes. What are there was a question about what are the good locations to go freshwater snorkeling to observe fish in their natural environment? Any recommendations? Or?
Yeah, many, I would say. Like, I don't know, maybe you should, if you can any place in the wet zone, like if it's a rainforest habitat area, like you can go to Cornelia, Singaraja, do some snorkeling, and also in the Knuckles range. Yeah, I mean, it's really it's more like what kind of species you would like to see. But if you really want to just get into water, do some snorkeling, just go out, take the highway to Atugama, Atweltoda, Singaraja, Kannel area, and you will find enough places to do snorkeling. Yeah.
Thank you, Hiranya. And I think with that, we can sort of wrap up for today. But I want to thank you for giving us a fantastic talk and for answering all these questions. The research you're doing is really special and it's outstanding. So we wish you from the WNPS, and I'm sure everyone who has joined the session today will wish you the very best as you pursue your PhD studies. And also, before we close, I would just like to remind everyone that Hiranya and Rohan Petiagoda have extended their pre-publication offer for the new book that is launching on the biogeography of Sri Lanka and on the freshwater fish especially. They're focusing on till the 21st of November. So thank you.
Thank you very much, Zainab, and thank you for providing me the opportunity to present some of the work. Yeah, thanks.
Thanks. Bye-bye.